Okay. Hi, everybody. This is Alicia Halliday. I see we have about 20 people on the line. Thank you for attending this Autism BrainNet webinar, which gives you the opportunity to learn about recent discoveries, recent research, things that are going on with autism brain tissue. You guys have all either participated as a registrant. If you haven't registered for the Autism BrainNet and you'd like to hear more about these webinars and all sorts of upcoming educational opportunities, you can do so by going to www.takesbrains.org and go to sign up, and you'll get a quarterly newsletter and be alerted about these opportunities. So I want to take an opportunity to introduce Dr. Christopher Walsh. Christopher Walsh is at the Boston Children's Hospital. He has both an MD and a PhD, so he has many hats at Boston Children's Hospital. But his lab is particularly interested in developmental disorders of the human brain, those that cause either epilepsy, intellectual disability, and/or autism. So he and his lab focus on genetics and molecular biology of the developing cerebral cortex, which is, of course, part of the brain. And he has the opportunity to discover, or at least participate in discovering, some of the more interesting types of mutations that are going on in the brain. And we thought it was particularly interesting or important to talk about these types of mutations called mosaic mutations because they are present in some brain cells, but not all brain cells. And so the brain is really the most crucial organ to understand these types of mutations in autism, but he's going to talk about a lot of different research that he's done. So without further ado, I'm going to hand it over to Chris Walsh. You should all be able to see his screen, which says at the top, mosaic mutations present in some brain cells, but not all brain cells, and their relevance to autism. Before we get started, just one housekeeping note. I know you're all going to have questions and want to ask further information of Dr. Walsh. You're all on mute, so you can't, so if you want to multitask, if you want to let your dog out, you can do all those things. You can go ahead in your GoToWebinar control panel at the very bottom. It says webcam, questions, polls, handouts, and chat. If you enter a message in there, I will be able to read it, and then I will collect them at the end of the webinar and then send them to Dr. Walsh or read them out for Dr. Walsh, and he can answer them. So there is a way for you to ask questions during the webinar. Without further ado, I'm going to hand it over to Dr. Walsh.
Good afternoon, everybody. I'm Chris Walsh, and thanks very much to Alicia for inviting me to do this webinar on this really important topic of how to best use the brains of kids who died with a diagnosis of autism and whose brains the families generously donated to try to help us understand more of the science of autism and what we might someday be able to do to try to improve the situation. I hope everybody can hear me okay. And I guess if not, let Alicia know and she can let me know as well. So I'll be talking about mosaic mutations. And these are mutations that are present in some cells of the body, but not all cells of the body. And I'll tell you a little bit about what they are and the relevance that we're thinking that they might have for autism spectrum disorders. And the term mosaic mutations isn't meant to imply that basically it's different from one cell to the next. There's another term called somatic mutation, which is used almost interchangeably. A somatic mutation means a mutation of the body, because soma means body. And these are also cells that are present in some cells of the body, but not all cells of the body. And the two words are more or less interchangeable. So sometimes I'll talk about mosaic mutations, sometimes I might say somatic mutations. And we're talking about somatic mutations that are clonal, which means they occurred in a dividing cell and created a clone of cells that all carry the same damaging mutation. And so they're clonal somatic mutations or mosaic mutations. And it turns out that we thought that every cell in our body had the same genome, but it turns out that in fact the genome of every single cell in our body is a little bit different, and I'll be telling you also a little bit about that over the course of the hour, or a little less than an hour. So I'll be telling you that these mosaic mutations can occur anytime a cell divides. It has to copy its DNA, and just like any kind of photocopier machine, anytime you copy something, the copy is never quite as good as the original. And that is the same is true of our genome. That has 6 billion letters of DNA, and they all have to be copied with complete fidelity, and they never are copied perfectly. And so every time a cell divides and copies its DNA, it looks like there are very, very few errors, maybe only 2 or 3 errors in the whole genome, but nonetheless, they can kind of build up, and every now and then one of these copying errors creates a clonal somatic mutation that happens to be damaging. Unfortunately, most of these mutations actually are silent. They occur in junk DNA or the parts of the DNA that doesn't encode for protein, but every now and then they can be damaging. And so I'll tell you that these clonal mosaic mutations can occur before or early in development, when actually the different tissues of the body and the different germ layers have not yet separated from each other. And that's illustrated by this pink cell. This blob of cells is the early embryo before it's even formed any pattern whatsoever. And this one pink cell has developed a mutation compared to all of the other normal gray cells. And then that mutation would be present throughout all the tissues of the body, the brain and the lung and everywhere else, in a fraction of the cells, maybe 5% of the cells or 10% of the cells. Or it can occur a little later. It can occur, say, after the brain cells, in this case, have separated from the lung cells and the skin cells. And so the mutation would then be present in a fraction of cells in the brain, but only in the brain and not in the blood cells, where we could easily detect them if we did the genome sequencing that we usually do on blood-derived DNA. And so we could only find these mutations by studying the brain itself, and that's where Autism BrainNet is so important. Or, in fact, it can occur even later, at later stages of brain development, so that it's not even everywhere in the brain. This great green mutation is present in a fraction of cells in one half of the brain, the left hemisphere, and not even in the right hemisphere. Or this blue mutation occurs later in the process of neurogenesis of the cortex, which refers to the formation of neurons. Neurogenesis means the birth of neurons, and cortical refers to the cerebral cortex, the biggest part of the brain that underlies a lot of our most human-specific behaviors like language and social behavior that can be so abnormal in autism. And so a mutation can occur anytime during this neurogenesis in the cortex. And so if it occurs early during that process, it can be present in maybe half of the cortex. If it occurs later, it can be present in a smaller and smaller fraction. Or in some cases, I'll tell you, non-clonal somatic mutations can occur after neurons have stopped dividing, because neurons in our brain don't usually divide. They're one of the few cells in our body that are post-mitotic, which means they no longer mitose or no longer multiply to form additional neurons. So for whatever reason, nature made the vast majority of the neurons in our brain not capable of division and not capable of regeneration. But, so sometimes it turns out these mutations can occur even after a neuron has stopped dividing, and so it's only present in one single neuron in the brain. And we can only detect that by the very newest technology that I'll tell you a little bit about. So we know that clonal somatic mutations are basically what cancer is all about. We've known for a long time that damaging mutations that arise in the lung as we age, and particularly if we're smokers, can cause cells to divide uncontrollably and can create lung cancer. Or mutations but those mutations only cause lung cancer and they don't cause cancer in the skin. Or if we get too much sun, we can get damage to our skin and we can get skin cancer, but that sun mutations, they don't cause lung cancer. And so these cancer mutations we've known for a long time, are mutations present in some part of the body, but not the whole body. And so I'll tell you that we now know that clonal somatic mutations cause certain forms of epilepsy, which are very important clinically and which can be treated by removing those mutated cells. And then I'll tell you that, and that's how we got interested in these clonal somatic mutations or mosaic mutations. And then I'll tell you about how we started thinking about whether clonal somatic mutations might be related to some kids on the autism spectrum. And finally, I'll tell you about how, in fact, studying these somatic mutations in single neurons tells us about how our normal brain changes with age. So first I'll tell you about epilepsy and clonal somatic mutations, and this is work of a former fellow by the name of Van Padori, who's an epilepsy specialist, and she was studying a disorder called hemimegalencephaly, an unpronounceable Greek word that means half of the brain. Hemi is way too big, meaning mega, and cephaly refers to the cortex. And so you might be able to appreciate that in these MRI scans of a patient here, patient number 3, where the normal brain is symmetrical. Here's the right hemisphere, which is shown on the left because the radiologists like to mess us up, and you see it's labeled right hemisphere, and the left hemisphere shown on the right side. They look pretty much the same. But in this patient, the right hemisphere is much bigger than the left hemisphere, and that's where the hemimegalencephaly comes in. And you see it's also kind of abnormally formed. The colors don't look quite right. And this image shows an image parallel to the brim of a hat. And you can see, again, the normal left hemisphere is completely outdistanced by this abnormally enlarged right hemisphere. And this child, this boy, from the day he was born, he started having intractable seizures, starting from this abnormally enlarged right hemisphere, because that brain is just a mess. It's completely disorganized. The cells are very abnormal. And the left side of his body, which is governed by the right side of the brain, was very weak because this right side of the brain wasn't working properly anyway. And so in a desperate attempt to control his seizures, his neurosurgeons took out that whole right hemisphere. And so now all you see there is this bright white stuff, which happens to be water or cerebrospinal fluid. And his whole right brain was taken out. And believe it or not, this was life-saving. He had no seizures for 6 years. He learned to walk. He developed fluent speech. He reads at grade level. And of course, he's still weak on the left side because he has no right brain at all. But in fact, his leg works pretty well. And with his parents' permission, I'm going to use his picture and name. His name is Dante, and he's an amazing kid. And this surgery, as dramatic as it is the only thing you can do for a lot of these kids that have intractable epilepsy. And Anne wanted to figure out what the heck was going on in Dante's brain, or in that half of Dante's brain, and wanted to understand if that was due to some mutation that might be present in the right brain, but not present in the left brain, which, as far as we can tell, seems to be working very well in him. And so, to make a long story short, because this work was published 5 years ago, we sequenced a gene called AKT3 in Dante's brain, and we found a very subtle abnormality in just one little part of that gene, and that's shown here by this. This is the DNA lettering, TAG, and so on. This is actually how, back in those days, we would monitor that DNA sequence with the different peaks in this chromatogram showing the different sequences. And this is what it looks like in Dante's blood, and that's what the normal sequence is supposed to look like. But when we looked at the sequence of the same gene in Dante's brain, there's just something a little bit abnormal, that at this one position, this one G, wasn't only G, there was a little bit of A in there, this red peak. It was mostly normal, but it looked like a fraction of the cells might carry a particular mutation at just that one place in the genome. And what seems to have happened in Dante's brain is that he's got this mosaic mutation. It occurred after the formation of the egg and the sperm, after they were united to form the zygote, and during embryonic development, one of those cell divisions created this spontaneous mutation that in Dante's case was only present in his brain, and in fact only in the right side of his brain. And not in all the cells of his brain, but only in a fraction of the cells of his brain. So even that abnormal part of the brain is a mix of good cells and bad cells. And so in fact it's a mixture. Outside of his brain, the cells are all homozygous, or in other words, they have 2 copies of the normal. They're homozygous normal or homozygous wild type, and that means they have 2 copies of the normal sequence, because as you know, we have 2 copies of all the genes in our body, except for those on the X chromosome, where males only have a single copy. But in Dante's case, a fraction of his cells had a good copy and a bad copy of the gene, and since that only a fraction of the cells carried this heterozygous mutation, that's why if you looked at the DNA from the whole half of the brain, this abnormal peak was a little lower than normal. And so this is a clonal mosaic mutation, and I'll tell you that the over the course of this talk. They occur in all of us. It's inevitable as cells divide that it'll happen every now and then. And hopefully the mutations that occur don't actually change the function of a gene anywhere. And in those cases, those are the majority of cases, it allows normal development of the brain. And so during the process of formation of the brain, the cerebral cortex is basically a large folded sheet, and it's formed from 6 layers of neurons that do various things, and those neurons are all shown by these little tree-like schematics here. And those neurons, in turn, are formed from dividing stem cells that are shown as these purple cells here down near the bottom. And they live in a stem cell zone deep in the brain called the ventricular zone. And then they form the post-mitotic neurons, and the neurons themselves, like I say, don't divide anymore, and they're stable throughout life. And in normal humans, the cerebral cortex is formed between 5 weeks of gestation, shown here by GW5, up to about 30 weeks of gestation. So it's actually more than half of the way through pregnancy. And in fact, there's actually some neurons that look like they're formed even up to the time of birth. And in one small part of the brain called the dentate gyrus, in fact, there are a few neurons that continue to be born after birth. But in the main part of the cerebral cortex, looks like most of the neurons are finished being formed by about 30 weeks of gestation. Now, this is much longer than in a mouse. In a mouse, the cortex is formed over about 1 week from embryonic day 11 to embryonic day 18, as opposed to these weeks of gestation. And the layers of the cortex are formed with the deep layers first and the upper layers last in the human as in the mouse. But the mouse, the whole process is much contracted because, of course, the brain is much smaller. And so what happens, oh, there we go, yeah. And so what happens in Dante's case is one of these stem cells got an abnormal mutation and then passed it on to to the daughter cells formed by that mother cell. And so a mixture of the neurons carried this abnormal mutation. And in fact, it's present only in about 1/3 of the neurons, but that's enough to create a complete, a big problem. Now, the particular mutation that Dante had is in a gene called AKT3, and it causes a substitution of the 17th amino acids from a glutamine to a lysine, or E17K. And we found, and others have found, that various mutations in AKT or in other genes can cause this overgrowth of the brain, or if the exact same mutation occurs in your shoulder, you can have overgrowth of the shoulder, or if it occurs in your legs, you can have overgrowth of the legs, or if it occurs in your big toe, you can have overgrowth of the big toe. And so these mutations are in a pathway called mTOR, which regulates the size of cells, and it regulates their proliferation and the identity of these cells. And basically these jazz up the whole system so that the cells hyperproliferate almost as they would in cancer, except they don't hyperproliferate so much to form an uncontrollable cancer, and especially in the brain because of the fact that the neurons actually have their own genetic program to stop dividing. They create too many neurons and abnormally formed neurons, but they don't create a continuous ongoing formation of too many cells as we would experience in a cancer sort of scenario. Now, hemimegalencephaly is rare. It occurs about 1 in a million people, but there's a smaller abnormality of the cortex called focal cortical dysplasia. So focal meaning a small spot, cortex referred to by cortical, and dysplasia just means an abnormal growth. And actually, these can create just a little patch of abnormal cells, about 1 centimeter or 2 in size, a single little fold in the cortex that's abnormal. And these happen to be the most common cause of intractable epilepsy in children that's so bad that it requires surgery in order to correct it. They can occur anywhere in the cortex, from the front of the cortex like here to the back of the cortex like here. They're much smaller, and these are 1,000 times more common than hemimegalencephaly, and might even be more common than that. As we get better ways of imaging the brain, we find these things occurring more and more frequently. And these are frequently then removed at the time of surgery. These happen to be also mutations in the exact same genes, but they occur later. So the hemimegalencephaly mutation would occur at this stage, so it's present throughout an entire hemisphere, but these focal cortical dysplasias happen later, so they involve a smaller patch of the brain and a smaller fraction of the cells, even within that abnormal patch. So this shows, for example, the exact same mutation that was found in 3 different lesions. These are 2 focal cortical dysplasias. This one's kind of hard to see, which is why I've surrounded it with a red circle. So you might have to take my word for it. This one's also sort of subtle, and so I've also surrounded it with a red circle. And these are 2 focal cortical dysplasias, and this is a hemimegaloencephaly, this time of the left hemisphere, which is shown on the right side. And in this case, there was a mutation present in only 3% of the cells, only from that piece of the brain when it was taken out at surgery, and not present in the blood or saliva of that same patient. And this was a mutation in a gene called mTOR itself, and it's a threonine at position 1977 of the protein swapped to make a lysine. And that exact same mutation was seen in another focal cortical dysplasia in about 3% of the cells. And then that exact same mutation was found in a case of hemimegaloencephaly, but in a larger fraction of cells, now almost 20% of the cells, and a broader extent of the cells, suggesting that that mutation occurred earlier during the process of brain development. Now, in recent work, we've tried to figure out what's the cell that's most important to causing the epilepsy. And in order to do that, we studied these focal cortical dysplasias, and we separated the neurons from the glial cells. And we asked, do you have to have a mutation in the neurons or in the glia? And then we separated single nuclei of those cells, and we amplified the genome of that single cell, and then sequenced the genome of single cells one at a time to find out which cells had the mutation and which cells didn't have the mutation. And we analyzed 3 cases of focal cortical dysplasia and 3 cases of hemimegaloencephaly. And as you would expect, the focal cortical dysplasias, since they're smaller, they carry the mutation in a smaller fraction of the cells, which is shown here on the y-axis. And the hemimegaloencephaly, since they're larger, they have the mutation in a larger number of cells. And then we measured the mutation in the total DNA from the brain, which is shown in purple, and then the DNA from neurons, shown in blue, and DNA from glial cells, which are negative for this neuronal marker called nu-n, and that's shown in light blue. And what we found is that the smallest lesions actually only had the mutation in the neurons and not in the glia, both of these cases. And then the larger lesions had the mutation in all of the different cells. Again, consistent with the idea that these small lesions occurred early in development. These small lesions occurred late in development, late enough so that the lineage leading to neurons and glia had already separated from each other, and that these guys occurred earlier in development at a stage when neurons and glia still had common stem cells. And what we find then is that it looks like if you just have the mutation in the neurons, that seems to be enough to create all sorts of mischief and enough to give you epilepsy, because these patients had epilepsy severe enough to undergo resection. And so it looks like the mutation in the neurons looks like it's the key to creating the epilepsy. And also we found that these 2 lesions are part of a continuum, fairly smoothly distributing, again, consistent with the idea that it's not the mutation that's important, it's exactly when and where it takes place. In all of these dysplasias, you see that the mutation's actually only in a fraction of cells, not in all of the cells, suggesting again that the normal and the abnormal cells are intermingling extensively. Oops. And this same point is illustrated in this slide, so I think I'll skip it. So next we wanted to ask the question, well, what about, could this explain some cases of autism spectrum disorders? As we know, some kids with autism are severely disabled in all spheres, but other kids on the autism spectrum actually are severely disabled for social behaviors, but actually can have average or even above average capabilities in other spheres. And also, like epilepsy, spontaneous mutations are an important source of risk for autism spectrum disorders. So this pie chart here shows my own view of at least some of the known causes of autism spectrum disorders, and the gray part shows the unknown part, which is still the greatest majority. And so we know there are Mendelian single gene disorders like Fragile X syndrome or tuberous sclerosis, and these actually frequently occur de novo. And what that means is that the parents don't have the mutation associated with the disease, but the child does. And so it's coming out of nowhere. That's what de novo refers to. Other kids on the autism spectrum have chromosome anomalies. These are almost always de novo. Other kids have copy number variants, which are deletions or duplications of a segment of a chromosome somewhere. These are also typically de novo. Other patients on the autism spectrum have coding region point mutations that change the sequence of a critical protein and make that protein no longer functional. And these, again, are frequently de novo. So we know that all of these causes of autism can be de novo, meaning they can occur in the sperm or the egg. But what if the same mutation occurred later? Could there be a child with an autism mutation in half of his brain and not the other half of his brain, such that maybe his math area works really, really well, but his social area works very poorly? And so we wanted to test this in the case of autism spectrum disorders. You can imagine, again, you might have a kid with autism that might only have a mutation in half of his brain, or even in a smaller fraction of his brain. Beyond that, you could wonder whether kids with ADHD or even normal people might have such damaging mutations in just a piece of their brain that might cause a much bigger problem if it were present in all of the cells of the body. And so first we wanted to do the easier test, which is to look for mutations occurring at an early stage that we could detect in the blood, because so many kids on the autism spectrum have already had blood sequencing of their exome, meaning all of the coding parts of their genome. And so, and then we wanted to test whether there might be a mutation associated with autism in part of the brain, but maybe not even in the blood at all. And we could only study those kinds of mutations by the sort of resource that the Autism BrainNet is creating. And so to look at the blood cells, a former postdoc by the name of Elaine Lim analyzed almost 5,000, actually more than 5,000 families. These are parents with their affected children, generally normal parents with an affected child, and all three of these people, that's what we refer to by a trio, had undergone whole exome sequencing. So we thought, well, maybe we could identify a fraction of these patients with de novo autism mutations that are present in their blood, but not present in all of the blood cells. Maybe they're only present in 20 or 30% of the blood cells, and the same might be true that they're also present in only 20 or 30% of the brain cells. And so we were able to look at basically all genes in the whole genome because they'd all undergone this exome sequencing. And so here's what we found. If you look at a heterozygous mutation, like I showed you in the case of Dante's brain, when the mutation is present in the heterozygous state, then we refer to that as a 50% allele fraction, meaning there's a good copy and a bad copy. So 50% of the analysis of the DNA sequence shows the mutation, and 50% shows a normal sequence. And so that gives us an allele fraction of 50-50. And most de novo mutations occur in the sperm or the egg, so all of the cells of the child carry that mutation, and so they show this 50% allele fraction. But Elaine identified that some of the mutations had this lower allele fraction. It looked like it was only about 25%, suggesting the mutation might be present in some, but not all of the cells of the body. And so these are our mosaic mutations that are present, that occurred before separation of brain from blood, but still after fertilization. And we found that these make an important contribution to autism spectrum disorders. Overall, about at least 7% of the de novo mutations appear to be post-zygotic, which is just yet another word for mosaic mutations. And most of these were not detected by the sorts of analyses that people had done before. Some of these autism genes that we found had previously been identified as ones that can cause autism if they're present in the germline, such as SCN2A. Others of these somatic mutated genes hadn't been reported as germline mutations before. And we think that, like mTOR and those things, there might be special autism mutations that only might cause autism if they're present in the mosaic or somatic form, because they might be lethal if they're present in all of the cells of the body. And our work was complemented by work that was published a little bit before ours from Jonathan Pevsner's lab at Johns Hopkins, which had very similar results. So these are some of the somatic mosaic genes we identified. KLF16, the ones shaded in red, are highly expressed in the brain, so we think that these are the ones that are most likely to be important. SCN2A is a familiar autism gene. Some of these others, like SMARCA4, had been implicated in intellectual disability before, but not in autism spectrum disorders. And we found actually that SMARCA4 mutations associated with autism spectrum disorders occurred in a particular part of the molecule. So this is a cartoon of the protein of SMARCA4, And all of these mutations up here had been identified in cancer, and they all are loss of function mutations. These mutations have been identified in a syndrome called Coffin-Cyrus syndrome, and they're generally not loss of function but missense mutations. And these are mutations in autism which are also missense mutations. They're not the cancer-like mutations, and they occur all jumbled, all packed in one little part of the protein. And so overall, work from our lab, as well as 3 or 4 other labs actually presented at about the same time, suggests that as many as 5 or 10% of kids that have likely causative autism mutations are these mosaic mutations that you can find in the blood, particularly if you look even more carefully than exome sequencing is able to look. Some estimates suggest involvement in as many as 5% of all autistic children, actually, although we're not sure of that number is. I think we'll need to get more data to be absolutely certain what fraction of autistic kids have these mosaic mutations. They can occur in familiar genes or in some new autism genes. And we also analyzed to see if kids that have mosaic mutations have higher IQ than kids that have germline mutations. And in fact, we did find that that was the case. In the Simon Simplex collection, for which very good IQ data is available, we found that the mosaic mutations were associated with significantly higher IQ than germline mutations overall. And so as we learn more about high-functioning autistics, it might be that some of these kids with high-functioning autism might have these mosaic mutations. And we have no idea whether mosaic mutations might be important in other psychiatric illnesses like schizophrenia or ADHD, but they're an intriguing hypothesis to at least consider for those other disorders. So actually work that was going on simultaneously that was actually published a little earlier looked at postmortem autism brains to see if we could find somatic mosaic mutations in children that had died with diagnosis on the autism spectrum. And at that time we found as many brains as we could, which was about 55, and then 9 other cases where autism was suspected. We sequenced those, and then also 51 matched neurotypical control brains at postmortem, and we didn't sequence the whole exome, but only the best validated, highly penetrant autism risk genes, of which there's about 80 at that time. But what we did this time, we sequenced those genes at a very, very high level of coverage, because this is important to detect mutations. If the mutation is only present in 1 out of 100 cells, you have to actually sequence the gene many, many, many times over to capture that fraction of cells that carry the mutation. And so this would be looking at very late mutations. And in that study, what we found was that overall, autism brains had as you might expect, had a larger proportion of these brains carried damaging mutations in known autism risk genes. So if we looked at silent genes that don't really change anything about the function of the gene, they're equally common in control brains, shown in blue, and autism brains, shown in red. If we look at mutations that alter the function of a protein, they're actually significantly more common in autism brains than in control brains. If we look at more deleterious mutations, in fact, they're even more greatly enriched in the autism brains than in the control brains. And if we look at the most severe type of mutation, which are loss of function, which completely remove the function of one copy of the protein, you see that they show the greatest enrichment in autism brains compared to normal brains. We found, in fact, a lot of the brains in this BrainNet collection actually carry mutations in known autism risk genes, which gives us a way of trying to look very directly at the human brain in the context of these known autism risk genes, things like ARID1B, SETD2, SCN1A. Many of the genes that we found were associated with autism plus epilepsy, and that's because a lot of these autistic kids happen to die of epilepsy, which of course is more of a threat to life than the diagnosis of autism alone. Most of these deleterious mutations were not mosaic. Most of them were germline, but about 10% of the deleterious mutations were these somatic mosaic type. And that's consistent so far with what we see with our analysis of blood DNA. And so, but what's great about these postmortem brains is that then you can just study the distribution of the mutation in the brain. Here's a mosaic mutation in an autism risk gene called SETD2. And if we look at DNA from the prefrontal cortex, you can see that the mutation is present in a fraction of cells, but not all of the cells. It's present in about a quarter of the cells, but most of the cells are normal. Very similar to that case of hemimegaloencephaly that I showed you in the case of Dante. And then, yet this mutation is absent from the cerebellum, and so it's only present in the cerebral cortex. So it makes us wonder, what's the minimal part of the brain that has to be involved in order to cause autism? We hope by studying these mosaic mutations, we can learn more about the parts of the brain that are key to the development of autism. We found a couple of other somatic mosaic mutations. An SCN1A mutation that disrupted a splice site. This was present both in the cortex and in the cerebellum. Another mutation in a gene called CACNA A1C. It was also present both in the cortex and in the cerebellum, so they didn't show this differential distribution that the SETD2 mutation caused that I just told you about. Also, we found that Fragile X repeat expansions can be non-uniform across the brain, so you can have a bigger repeat in some parts in the cortex than in the cerebellum, which also might create some of the variability that we see in the manifestations of Fragile X syndrome in kids with that disease. So I hope I've convinced you that somatic mosaic mutations can cause some brain diseases. Some mosaic mutations involve brain and other tissues, but still cause mostly a brain phenotype. We don't know how common these brain-only somatic diseases are. We know they're important in epilepsy. We don't yet know overall how common they are in autism, and we don't know what other kinds of conditions might also show these somatic mutations. We know that mutations in only a fraction of cells can sometimes cause a severe disease. So mutations in a 10% or a quarter of the cells in a hemisphere can cause hemimegaloencephaly, an overgrowth of a whole half of the brain. FCDs have a mutation in only 1 to 10% of the cells. And so the topographic extent and the percent of the mosaicism of mutation seems to relate to the timing during which the mutation occurs during the process of development. So we don't know yet what's the smallest fraction of neurons that can be mutated to cause a phenotype, because neurons are so highly interconnected. They're like a network. And so if you had a mutation just in one cell out of a whole network, would that be enough to cripple that entire network? We also don't know the extent to which somatic mosaic mutations normally shape the brain of neurotypical individuals. So at the same time, we were trying to understand just how much does the genome of one neuron differ from the genome of the neuron next to it in a normal brain. So we know that sometimes damaging mutations can take place during normal development, but to what extent is the genome of this cell the same or different than the genome of the cell next to it? There might be retrotransposon insertions, single nucleotide mutations, all sorts of different things. And so we wonder, the brain appears to be normally some sort of mosaic patchwork of somatic mutations, but what are the numbers here? How many mutations, how often do they occur, and how often are they damaging? And so two really terrific students developed the method I already outlined, where we separate single neurons and put one neuron each into each one of these wells of a microtiter plate. You can then lyse the nuclei and amplify the whole genome using this specialized DNA polymerase called Phi29, and then you can do a whole genome sequence, but of the genome of a single cell. And this just shows the labeling of the neurons, and these would be the labeling of glial cells, and so we can separate the neurons from the glial cells. So I'll tell you briefly about what we've learned about the genome of a single neuron. We know that there are jumping genes in our genome called retrotransposons, and that these are occasionally activated during the process of neurogenesis, and so they can be spontaneous jumping of retrotransposon genes can be seen at a consistent but low frequency in human cerebrocortical neurons. So we find that 1 in every 2 or 3 neurons will have undergone one of these retrotransposon jumping events. We also found that a typical neuron will have a large deletion or duplication somewhere in its genome, about a megabase in size or larger. And so these are a very important source of diversity of neurons, because really these large CNVs frequently alter the function of genes, because they'll typically duplicate or remove anywhere from 5 to 30 or more genes. And so this is remarkable that the genome of neurons can be unique in such a large and potentially functionally important way. And then finally, we found that, in fact, a typical neuron has about 1,000 point mutations that differentiate it from the neuron next to it. Now, most of these don't alter protein function because the coding part of our genome is about 1% of the genome. So if you divide this by 100, it will be about 10 potentially function-altering mutations, and then 90 compared to 90% of the mutations that are just silent because they occur in unimportant or non-functional parts of the genome. But we found that some of these are clonal and developmental, and some of them are only present apparently in a single neuron. These mutations, actually, the silent mutations form a lineage map, and so that we took a bunch of neurons. Every like black line corresponds to a single neuron, and then these colors represent somatic clonal mosaic mutations, and we can actually organize these neurons that are all from the same patch of the dorsolateral prefrontal cortex. We took thousands of cells from the dorsolateral prefrontal cortex, we sorted them into single cells, we analyzed their genomes, and we found that, in fact, these neurons from the prefrontal cortex form a family tree, so that these cells all share the same somatic mutation, and so they're all sister cells. These cells all share the purple mutations, and that there are 2 purple mutations. This one must have occurred earlier than that one because this one's present in all of the cells in this little clade or lineage, and this was present in some but not all. And then you see that there's very complex patterns of cell lineage. This C1 would be a mutation that occurred very early, and then these later higher number Cs occurred progressively later in development because they're only in a small fraction of the cells that are in that total big family. And so, in fact, all of us carry in our brains a permanent map of where all of the cells came from. In fact, our whole body carries this permanent map of where every single cell came from that can be, in principle, reconstructed post-mortem. And if we look at this particular branch of the lineage here, shown in blue, we can see that these early mutations, C1 and C2, occurred very early. They're present in up to 1/4 of the cells. They're in both halves of the brain. They're all throughout the brain, and in fact, they're also in a lot of these are ones that occurred before brain tissue separated from other tissues of the body. And then these mutations are like Dante's. They're present in a fraction of the cells and only in one hemisphere and not the other hemisphere. And then these mutations occurred progressively later. So you see that they occur in a smaller and smaller part of the brain and also a smaller and smaller fraction of the cells within that small area. I'll skip this in the interest of time because I think we're getting a little bit short on time, but I'll just summarize to say that we found that the structure of the cortex is much more complicated than we thought, because we could find these mutations that occurred very early during development that actually spread to the brain and to the heart and to the liver. And so this pink grandmother cell gave rise to cells in the heart and the brain and the liver, and then the daughter cells of this pink grandmother cell are intermingled with daughter cells of this blue cell and this purple cell, so that a given column of the cortex actually is very diverse in its clonal origins, tracing all the way back to multiple precursor cells present at the earliest stages of development. Now, in the last 5 or 10 minutes, maybe 5 minutes, I'll try to do it in 5 minutes, I'll tell you a little bit about what we've learned about mutations that occur after neurons stop dividing as they age, because some of these somatic or mosaic mutations are shared by multiple cells because they occur in stem cells during the development of the brain, and other of these somatic mosaic mutations are actually unique to a single neuron because they occurred actually after the neuron became post-mitotic. So it turns out that as we get older, our genome gets a little damaged, and we wanted to understand how, to see if we could understand more about what to do about that. And so this is a study that's now just appearing in Science. It should come out in print. It's already available online. We looked at not only the dorsolateral prefrontal cortex, which is our standard, but we then compared it to the dentate gyrus of the hippocampus, because this continues to form neurons after birth, and so the cells are continuing to divide, so we thought that they might be more likely to be damaged by these mosaic mutations, and also because the temporal lobe is so important in the genesis of Alzheimer's disease, which, as you can see from this PET study, starts first in the temporal lobe and then spreads more slowly to other parts of the brain. And so we basically looked at newborn brains all the way up to old age, and we isolated cells from the prefrontal cortex as well as the dentate gyrus of the hippocampus, and we did the same single nucleus isolation protocol, and then we sequenced the genomes of the cells and figured out how many point mutations are present. And we sequenced basically just a couple of cells from each place, 3 or maybe a dozen single neurons, and then we took sort of the average to determine the overall average for that person. And what we found, somewhat much to our chagrin, was, at least much to the chagrin of someone like me who's kind of getting older, is that when in the brains of newborns, the number of mutations was around somewhere between 300 to 900. And so the light blue shows one newborn who is 4 months old. The darker blue shows another newborn who's 6 months old. And you can see that their genomes look pretty good. And then we looked at several teenagers, and they had rates that were overlapping but a little bit higher. And then we looked at some middle-aged folks, and again, their numbers were overlapping but pushing a little higher. And then there's this one neuron that looked very sick and had thousands of mutations, over 7,000 mutations. And then finally, we looked at the elderly folks, and their mutation count per cell was consistently higher. So here are 4 cells, or 3 cells, 1, 2, 3, all from the same person. Here's 3 cells from another person. Here's 3 cells from another person. What you see is that within a given person, one neuron's a little different from the next, but you can get an overall average. Like I say, occasionally you see these outliers, but if you take the overall average, in fact, there's a slow, inexorable gain of mutations, about 1 every 2 weeks throughout life, so that you start at, so by the time you're in your 70s, a typical neuron will have over 2,000 mutations in the genome. And as if that isn't bad enough, the dentate gyrus of the hippocampus, probably because of that cell division, accumulates the mutations even faster, and so that the elderly folks in their dentate gyrus had up to 4,000, 5,000 mutations per genome in those cells. We also found that different people at the same age age a little differently. So you see here's a middle-aged person whose mutation count looks much more like the teenagers. And so other than the fact that she's dead, she looked like she was doing really well in terms of healthy aging. And so that implies that maybe either our underlying genotype or maybe some aspect of environmental exposures might be important at regulating this accumulation of mutation with age. We also looked at 2 diseases where there's premature aging. Kids get gray hair when they're 5 years old, and they get premature degeneration. We found that, in fact, this premature neurological degeneration is associated with accumulation of mutations at a faster rate. We looked at Cockayne syndrome and xeroderma pigmentosa. These are disorders actually of DNA repair. And so it had already been suggested that this abnormal DNA repair might cause an accumulation of mutations at a young age, and that might be important for the neurological degeneration. We found that both Cockayne's and xeroderma pigmentosa had 2 or 3 times as many mutations as single neurons from normal people of the same age. And so I'll just close by giving you our present model for how we try to understand this tremendous mosaicism that affects the human brain. So when a zygote is formed, it already carries about 100 de novo mutations that are present in the zygote and not present in either parent. These are the mutations that occur in the sperm or the egg, and only about 1 of these affects the coding part of the genome, that is the exome, fortunately. And so most of these are not functional. These are rarely functional, but when functional might cause de novo disease, such as autism. As I say, 99% of these are functionally silent. And then that zygote divides, and it divides again. And every time it divides, it looks like there are 2 or 3 point mutations that occur every time a cell divides. Plus, occasionally you'll have a copy number variant or a retrotransposon or a microsatellite mutation. And then once you have a blastocyst, you find these different lineages then are distributing to different tissues. By the time you have a fixed brain, and by the time the child is born, there are somewhere between 300 and 800 unique mutations not present in the parent or the zygote in that single neuron, and then in a different neuron, a different suite of unique mutations that are not present in the parent or in the zygote. Now, Again, most of these are functionally silent and fortunately don't impair the function of the neuron. But many neurons will have accumulated at least one clonal damaging SNV by the end of neurogenesis. But fortunately, different neurons carry different problems, and so that seems to allow them to work together reasonably well. And healthy people presumably avoid deleterious mutations that occur early on in development. I think I'll skip this slide because I think my time is running a little short. And then after birth, somatic mutations that are unique to the single neuron start accumulating with age. As I've outlined already, one mutation every 2 weeks or so in the frontal cortex, one mutation every 10 days or so in the case of the dentate gyrus of the hippocampus. And so that by the time we reach our 70s or 80s, our genomes are sort of looking like these, in terms of the mutation count, are in the same range as these neurological disorders that are associated with neuronal degeneration and abnormal function. And so do these accumulating mutations contribute to age-related cognitive dysfunction or common forms of neurodegeneration? Well, this is a huge question mark, and this is obviously what we would like to understand as soon as possible. We'd also like to understand, is there more that we can do to try to prevent these mutations? Our evidence that I won't go through suggests that at least some of these mutations reflect abnormal oxidation, and so that's got me eating a lot of blueberries these days. But would like to know more about what kind of oxidation and what causes that oxidation and what might be able to do to prevent it. Finally, we're very grateful for using the Autism BrainNet project. This allows us to do a wider range of analysis of postmortem autism brains, and we're doing high coverage sequencing of the entire genome to be able to look systematically at the entire pattern of mosaic mutation in these autism brains using a whole genome sequencing at a very high coverage. And as I say, this shows the pattern of somatic mosaic mutations in one normal brain, and these would be mutations. Oops. These would be mutations present in all cells, which actually there's lots of them there, but we filtered them out. These are mutations present in half the cells, present in 25% of the cells, and you see that a normal brain contains lots of these somatic mosaic mutations. Fortunately, most of them don't seem to alter function. Here's an example of an autism brain. This is data hot off the press from a student by the name of Rachel Roden with help from Peter Park's lab and Max Sherman. And this is a patient that has a somatic mosaic copy number variant that's present in some but not all cells of the body. And you see it's on chromosome 2, it's about 7 megabases in size. It involves a region of the genome that had previously been implicated as an autism risk region of the genome. So we think this is probably causative, but in fact it's only present. And you can see that in control brains, you're supposed to have 2 copies of this segment of the genome. This patient has closer to 3 copies, but not quite 3 copies. We think it's actually a very complex rearrangement. That's actually a that's actually a very complex rearrangement that involves more than just a duplication, but only in about 20% of the cells in the brain. So just to conclude, one brain has many genomes. The development of each human brain is a unique history, with these clonal somatic mutations representing a durable mark of the pattern of cell divisions. These clonal somatic mutations cause a fraction of autism. We don't yet know exactly how high of a fraction, and we don't know about their role in other disorders. And then finally, somatic mutations occur universally in the normal brain. And we just have lucky people who don't seem to have too many damaging mutations, and unlucky people who are not so lucky. So thanks for your attention. I mentioned the work of many people in my lab, but I didn't mention, I mentioned Anne. Salmia's work I didn't talk about. Alyssa did the focal cortical dysplasia work. Mike Lodato, Molly Woodworth, Michael Coulter, Rachel Reef-Rodan did the single cell analysis and the aging study, and that was done very closely with a collaboration from a superb lab run by Peter Park, Craig Borson, Allison Barton, Daniel Kwan, and Alice Lee. Thanks very much for your attention, and I'd be happy to answer any questions if there are any.
There are some questions, Dr. Walsh, thank you. There are some pretty technical questions that I assume are coming from other scientists that have to do with different types of protocols that were used. So I'm going to skip over those, and if anyone has any specific questions, you can reach out to me directly, and I'll coordinate with Dr. Walsh. But there were some questions. A lot of them also, just to be clear, we don't have copies of the slides, but absolutely this is being recorded, and the recording will be available on autismbrainnet.org probably by the end of the week. So don't worry about having the handouts. You're going to have the full presentation available on a static site for you to view and review as you would like. So we do have a couple questions. One is, do you think there's any significance to the fact that tuberous sclerosis TSC mutations also produce disruptions in mTOR signaling?
Oh, absolutely. So I mentioned, I did not mention it, but the TSC is, as the questioner undoubtedly knows, a negative regulator of the mTOR pathway, and we found that some of these focal cortical dysplasias are actually due to TSC mutations. They sometimes occur in patients that actually don't carry a diagnosis of tuberous sclerosis. They don't have the skin lesions, they don't have the heart lesions, and in fact, some of these patients really and truly don't have tuberous sclerosis. A couple of our patients with hemimegalencephaly had tuberous sclerosis mutations and had been seen at some of the leading tuberous sclerosis centers in the country, and actually found not to have any of the diagnostic criteria for tuberous sclerosis. And that's a weird thing. For some reason, it makes us think that, in fact, tuberous sclerosis related lesions are more common than we realize, and that tuberous sclerosis has a variability of penetrance that we hadn't previously appreciated.
We have another question about premutation carriers. So is there a way to test if a premutation carrier actually has a mosaic mutation?
The Fragile X premutation, you mean?
This person didn't state specifically, but I'm assuming that's what he or she meant.
Yeah. So, well, basically, we in our finding that the premutation carriers of Fragile X had the full amplification of the repeat in their brain, but not in their blood. We used the standard Fragile X testing protocol, but of course we had the postmortem brain available, and so one might be able to get some sense of that by studying the blood and looking specifically to see if there might be a tiny amount of amplification in the blood that would make it more likely. But I'm not sure that there's another way of doing that short of having some sort of brain tissue available.
Okay, and there's one last question. I apologize. There was a question from someone about the interplay between point mutations and somatic mutations. However, I'm going to defer on that one. So this is a question I think that's come up a couple of times. So if mosaic brain mutations or mosaic mutations that are seen in the brain but not in blood can't be detected in the blood, how can you or do you see a possibility of a test for this in the near future?
Well, the only way we could, well, obviously, we can't do it definitively without getting brain tissue. And so that's, again, where it's important to understand. That's why the Autism BrainNet collection is so critical, because it allows us to evaluate the hypothesis of how important are these things. And so exactly how much are we missing? Do we need to develop such a diagnostic? The epilepsy ones, they can't be diagnosed. They are typically only in the brain and not in the blood, as I showed, because of the fact that they cause this overgrowth phenomenon, and so they can't really be diagnosed other than by removing the tissue. And we're trying to, on the epilepsy side, we're trying to learn more about whether the different mutations can help us understand more about surgical outcome and things like that. But that's really, down the line. It's possible that some people are interested in seeing what sorts of DNA might be, in the cerebrospinal fluid, or whether other parts of the body, maybe, the skin, for example, is more closely related developmentally to the brain than is the blood. And so some of these brain mutations are more likely to be represented in skin tissue or hair tissue than they would be in brain. So it's possible that for some cases that might be useful, but I think for the time being, we don't have a good way of diagnosing them.
Well, thank you again so much. We're a couple minutes after. If someone didn't get their question answered and they still have it, they can email me at Halliday, AHALLADAY, at autismsciencefoundation.org. My email address is in the chat box. Again, the presentation will be is being recorded and will be posted on autismbrainnet.org in about the next week. And we really, really thank you for participating. Thank you to Dr. Walsh for making this presentation. And thanks to all of you who attended and stayed. Most people stayed, which is really a unique experience. So thank you all so much. And if you haven't already registered for the Autism BrainNet, you can go to www.takesbrains.org and click register. So thank you so much, and we look forward to the next webinar.
All right, thanks all very much.
Thank you.